Properties of different yeast vectors. Now, from our recent discussions, we have the idea that number of plasmid vectors they are available for cloning or manipulation for yeast cells. And I have quoted a few examples that again we are going to elaborate them that why they are so much desirable to perform cloning in yeast cells. So the availability of such variety of plasmids have provided a lot of options for the geneticist to perform gene manipulation or cloning procedures by using yeast cells. As I told you earlier that plasmid vectors that have the ability to replicate in two diverse hosts like in E. coli or yeast cell, they are much more favored because in case of E. coli हम ये कर सकते हैं कि ज़्यादा से ज़्यादा कॉपीज बना सकते हैं रिकॉम्बिनेंट प्लाज्मिड्स की जिसमें हमारा टारगेट जीन भी रेप्लिकेट करता है क्योंकि ई कोलाई के अंदर इसकी जो रेप्लिकेशन है वो कंपैरेटिवली इजी है हम ज़्यादा क्लोन डेवलप कर सकते हैं और जब हमारे पास बहुत सारे क्लोन आ जाते हैं तो फिर हम उनको यूज़ कर सकते हैं यीस्ट सेल्स को ट्रांसफॉर्म करने में। तो इस तरह से बहुत सारी जीन्स जो हैं वो ऐसे फंक्शन भी परफॉर्म करती हैं जो कि कॉम्प्लीमेंट करती हैं सम म्यूटेशन एनालिसिस में। कॉम्प्लीमेंट का मतलब है कि कुछ ऐसे म्यूटेशन एक्सपेरिमेंट्स हम परफॉर्म कर सकते हैं जिसमें हम ऐसी जीन जो हमने म्यूटेट करवाई है उसको हम कॉम्प्लीमेंट कर सकते हैं या उसकी जो वाइल्ड टाइप फिनोटाइप है उसको हम रिस्टोर कर सकते हैं। तो यीस्ट के साथ जब हम एक्सपेरिमेंट करते हैं तो लाइक बैक्टीरिया हमें ये ख्याल रखना पड़ता है कि क्या उसका कॉपी नंबर जो है वो मॉडरेट है, ज़्यादा कम या बहुत ज़्यादा तो नहीं है क्योंकि अगर कॉपी नंबर बहुत ज़्यादा हो जाए और अगर हम उसमें कोई ऐसी जीन एक्सप्रेस करवा दें जो कि सेल के लिए टॉक्सिक हो लाइक एक्टिन की जीन्स या ट्यूब्यूलिन जीन तो उससे सेल की जो है वो फिजियोलॉजी डिसरप्ट हो सकती है। सो ऑल दीज थिंग्स वी शुड कंसिडर दैट what is the frequency of transformants by using a specific type of plasmid vector or what is its stability, whether they are stable after transformation or not. And copy number, of course, it is very important because if copy number it is high or moderate, then the gene that has been recombined or transformed within the yeast cell, it is easy to recover after cloning procedure. Again, if we focus on different examples like YLP plasmids, although its transformation rate it is low, like 10^2 transformants can be obtained by using per microgram of DNA, but more or less it is stable. On the other hand, yeast episomal plasmid vectors, they are having high transformation frequency that may be up to 10^5 cells by using per microgram of DNA. And we should consider here that toxic gene expression should be minimized if we are going to express such genes that can disrupt the host physiology. But high copy number, it is having the advantage that its recovery is very easy. On the other hand, the most favored one, it is the yeast centromere plasmid or YCPs because its copy number is maintained at moderate level that may be 1 to 20 copies per cell. And it is stably maintained, so it is much more favored. Although variety of examples may be there, but we can quote a few one in next table. This table, it contains different properties of yeast vectors. So the first example is YLP vector. its transformation frequency, it is low, around 10 raised power 2 transformants per microgram of DNA, but copy number, it is very, very low, but it is stably maintained. Then yeast episomal plasmid, its transformation frequency is high, up to 10 raised power 5 cells, and copy number, it is between 25 to 200, very high copy number. And yeast replicating plasmid, its transformation frequency, it is 10 raised power 4, and copy number is moderate, like 1 to 20. And the most favored one, it is the yeast centromere plasmid, because it is also having high transformation efficiency, but copy number is low, but it is very important to stably maintain the trans or the recombinant DNA molecule. And of course, the yeast artificial chromosome, it is very famous, that can be used to clone high or the large size DNA molecule, and copy number, it is 1 to 2 per cell. So these are variety of examples of vectors that can be used for cloning, but in addition to that, there are some other examples that depend that what procedure we are going to perform with the yeast cells.